Hello, good morning, Prof. From another. Dear Prof, I think we can hear.
Yeah, fine. Good morning.
Good morning, Prof. Nice to have you here, Prof.
Thank you very much. Thank you.
Welcome, Prof. Sorry,
Yeah, welcome.
the early morning, Prof, because I think it starts at 10 o'clock, right?
Yeah. No problem. No problem.
We shall wait for a few more minutes, Prof, because the people will join, I think.
Yeah. Of course.
Maybe we should wait for another 2 more minutes or so that the participants will join. Please bear with us. So how is the situation back in India, Prof? Fine?
Yeah, it's improved, and I think Delhi, it's a lot of improvement.
Okay, great.
Now I think maybe next month everything will start slowly. I think Malaysia is it's increasing.
Yes, exactly, Prof. The cases are keep on increasing every single day, even after the lockdown and so on. But I think it will recover better. Another 1 or 2 more weeks to go.
2 more weeks, okay. Because my wife and everybody is there.
Okay.
And
Yeah,
they struck there.
I see.
They came to renew the passport. Hmm. Hmm.
I think it is better to start. It seems maybe the participants will join while
Okay.
the presentation
Okay.
is on.
Hmm. Okay.
Bismillah ar-Rahman ar-Rahim. Alhamdulillah Rabbil Alameen. Let us start our webinar with Surah Al-Fatiha. Respected presenters and dear participants, I welcome you all with Islamic greetings. Assalamu alaikum wa rahmatullahi wa barakatuhu to our yet another webinar powered by Institute of Oceanography and Maritime Studies and Innocent Marine School, which is the flagship program of our International Islamic University Malaysia. We used to conduct the workshops and the seminars every week, every month, and this is in conjunction with the World Ocean Day, which is on June 8th. But we couldn't conduct it on exactly on June 8th because of a lot of programs all over. Uh, all the other universities were conducting as well. So we plan to postpone to this date, June 11th, and we invited two presenters this time, and one is from India, Professor Ramanathan. He is a full professor from Jawaharlal Nehru University, one of the leading universities in India. Another one, the second presenter would be Dr. Najat from our Marine Science Department, from our faculty itself. For the smooth running of this program, I would like to request the first presenter would be Dr. Najat, followed by Professor Ramanathan. And let me introduce the first presenter. Dr. Najat, she is our faculty member, and she has secured her PhD in the bioscience, the field of bioscience, and master's in bioscience as well from same International Islamic University Malaysia, and her bachelor is on biotechnology. She is currently an associate professor and serving Faculty of Marine Science, Department of Marine Science and Faculty of Science, International Islamic University Malaysia, and she will be presenting an interesting topic which everyone will enjoy for sure. And she'll be presenting it for about 20 to 25 minutes, followed by a question and answer session, and followed by Professor Ramanathan, who will take over the floor. Without further, I would like to invite Dr. Najat to take over the floor. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much, Dr. Akbar, for the very sweet introduction. And thank you everyone else who are here willing to spend some time of your busy schedule to join this webinar. I'm honored to be invited in this webinar and speak on the research topic that I did for my PhD recently that I finished in this webinar today. Which the theme that we have for this webinar is Ocean Day. I believe the theme is in line with the celebration of World Ocean Day, which was a few days ago this week. And also with the launch of Ocean Decade Initiative by the UNESCO. So my research topic was related. I gave the title for today's sharing, the bacteria workers in a seaweed factory. I like to use these catchy terms, bacteria workers, seaweed factory, for like my previous 3MT, 3-minute thesis, and other related presentation, like euphemisms. Reason being that my research was on the symbiotic bacteria that exist in the seaweed samples that I took, which in this case was Bacillarium mainensis and Lawrencea sp. This bacteria plays an important role in the seaweed ecosystem. So it's like the importance of workers in a factory. So I forgot, I wanted to share my screen. Is it possible? Just
Please do share the screen, Doctor.
it's here, okay.
Perfect.
So to start off, the red seaweeds that I took in my study, they were Gracilaria maninensis, which is an edible seaweed, which I took from two settings, one from a farm culture setting and one from a natural setting, the wild. And then I also took Laurencia sp from a natural setting. So these are used for comparison now. The idea was for me to take bacteria from the farm culture was to identify what bacteria is helpful in the farm culture for seaweeds. So these red seaweeds are categorized under Rhodophyta, which is red, given for the pigments of the seaweed, phycoerythrin and the phycocyanin. But sometimes these red seaweeds can appear greenish or brownish because sometimes the pigments of the phycoerythrin, phycocyanin is lesser than the other pigments that can exist in the seaweed, such as the xanthocyanins, giving those greenish or reddish, brownish colors. Red seaweeds are is a very large group, have more than 7,000 species, and they are commercially important. Most of those species are commercially important. For example, the Coprophycus, which is often harvested for carrageenan, and the Gelidium, harvested for agar, and the Porphyra, which is also another example of an edible seaweed, which is used in the Japanese cuisine as nori, the seaweeds that usually which appears green when you eat it, but it's actually from the red seaweeds also. So farming is important. Farming of seaweed is done globally. So knowing how to farm efficiently is like a factory. So hence there's bacteria workers in the seaweed factory. To introduce the seaweed ecosystem is very important to study because much is needed to be known for especially to know efficient farming. More often the study on seaweeds is more on cultivation, which is true. Cultivation methods are always important if you want to study the seaweed. But also biotic factors such as the microscopic symbionts that exist in the seaweed system is also needed to be known. So my studies the seaweed in this studies the bacteria in the seaweed. I apologize. Seaweed symbiosis. Because I'm sorry, she has to come in. Because in the holobiome, what is meant by holobiome here is the ecosystem by which the host and other species exist simultaneously, and they form one ecological unit. So in the sense that the seaweed, it has as a host here, and there are resident bacteria and also other microscopic beings such as the virus, the bacteriophage, and the fungi. So other microscopic beings are also there. So what the host provides for the symbionts is that oxygen, shelter, protection against predation, and harmful radiation. So in return, these symbionts, for which in my study I studied bacteria, they provide back to the host the carbon dioxide, the fixed nitrogen, the vitamins, the minerals, the detoxifiers, and the antifouling compounds, etc. So what I did in my study, well, I did the sample collection, I did the species identification, I did the culture independent approach to identify the bacteria species, and also the culture dependent approach. I applied the NGS, which is next generation sequencing, which enables the multiple sequencing of genes. Multiple sequencing of gene, which is much faster than Sanger sequencing. But however, I did also do the traditional culture, which employs the Sanger sequencing. The NGS is able to amplify multiple genes at the same time. Give me a moment. I'm so sorry. I apologize for a moment.
It's fine, doctor.
So those are the perils of working from home. I apologize. Okay, back to what I was trying to say. I employed NGS as part of my research method with also the traditional culture. So I also employed the 10D sequencing. The advantage of NGS because it's faster, it can multiply the sequences with much faster efficiency. And in that case, I was able to identify more, I obtain more reads, which is deciphered into more taxa. So NGS is useful for biodiversity study or the studies of the phylogeny of uncultivable microbes because the big plate anomaly. The big plate anomaly theory says that there's only like 5% or 10% of the bacteria that is able to be cultured. There's much more microbes that cannot be cultured, but is yet to be identified in the environment. That's the belief. So the advantage of using, for me in my research, to do both methods, I get to see that by amplicon sequencing, which is NGS, under NGS there's multiple techniques. So the technique that I use is the amplicon sequencing for next generation sequencing. The amplicon sequencing for me reveal more bacteria phyla than the cultivar isolates. For example, in my results, I got cyanobacteria, which in a large amount or a large percentage, followed by proteobacteria, followed by phytomycetes, Firmicutes, Bacteroides, Actinomycetes, Bacteria, Chloroflexa, I have no idea how I'm supposed to pronounce it. Gematimonidates, I believe. So these phyla actually they're under the 11 marine phyla that are common in the marine environment. However, for the cultivar method, I am only able to isolate and identify the four phyla, which is Proteobacter, the Firmicutes, Bacteroides, and the Actinobacteria. And also from the Proteobacter, if I compare between the amplicon sequencing data and my cultibacteria data, the cultibacteria only has like 2 subphylums of Proteobacteria, which is the alpha and the gamma. Yet the NGS data, the amplicon sequencing data showed more subphylum for the Proteobacteria, which includes the alpha Proteobacteria, the beta Proteobacteria, the delta Proteobacteria, gamma, and also there's a one species taxon under the epsilon Proteobacteria. So yes, for phylogeny or purposes or for like biodiversity, where you want to see the overall picture of these bacteria, like who exists in the marine environment, NGS is a very good way of doing it. However, by also employing the traditional culture method, I was able to use the selective enrichment media to promote growth of potentially new novel but bacterial isolates, because these media have supplements nutrients that is required to support biological profile of bacteria, biochemical profile of bacteria. It's about because some bacteria they need different glucose or different carbon or nitrogen source, such as glucose or glycerol. There's a difference between using glucose and glycerol also determines the bacteria that grows. The difference between using ammonia or nitrate, supplying a different form of ammonia or nitrate, also influence the type of bacteria that goes on the media. Introducing trace elements such as heavy metals. Sometimes some media are customized to place in heavy metals to see if the bacteria grows on those media. Some media are supplemented with vitamins and ions and salts. There's also like complex ingredients such as the yeast extract and the casein hydrolysates. This is all to promote the growth of the bacteria in sense of trying to see if these bacteria use different type of carbon source or different type of nutrients to grow. And some bacteria, they do use different, so sometimes when you just test, okay, let's try using halogen ions, or sometimes just try using different ions. Using these ions I'm sorry again. Using these ions will let the growth of some of these bacteria compared to other bacteria. And when you do the NGS, you usually want to assign those bacteria, assign your taxa according to the 95-97% similarity. However, when we do the this culture dependent method, when there's a bacteria isolates and it shows like good properties or like interesting properties like color pigments, the yellow, the blue, the green, the red, the black, or sometimes they show agar degrading properties, or sometimes they show other morphologies which are interesting as a microbiologist. We can use these isolates. We can identify the isolates based on the 16S. And if the isolates are not like 95 or 97% similarity, they are like 85% or 90% similarity. Usually we can look back into their genes and see if the sequence is nice. So if those gene sequence are good, yet the similarity is lower, like less than 95%, then we can like try see these as being novel bacteria. So in my study, I also saw the effect of salinity and other biotic factors on bacteria diversity. So obviously when we study any environment, such as the seaweed environment, the bacteria environment, the water environment, we always go include our data to be compared with these physical chemical parameters such as the pH, the temperature, the total suspended solids, the nutrients such as dissolved oxygen and nitrate. But in my study, salinity was a very huge factor in showing different bacterial diversity. But other factors that could contribute, which was not part of my study, was also like host species phylogeny and also biography and environment. The environment that the seaweed is in, either it's being in a farm culture or in a natural setting. If the setting, the farm culture setting or the natural setting is close to open river or close to agricultural farms, other agricultural farms, then those are factors that can affect bacteria diversity. And so for my when I did after I did the NGS and I did the phylogeny of my bacteria and I analyzed the nearest neighbor. So I saw that the nearest neighbors, they are grouping. I can see groupings of nearest neighbors being from the marine holobrine cluster or the seaweed holobrine cluster. This is to say that the seaweed, the bacteria that I found were closely similar to other bacteria that is found in other seaweeds or other marine hosts, such as sponges or corals. And I also did the functional annotation of those bacteria, because the bacteria that I found were from phylum Proteobacteria, the Prometheus, the Echinobacteria, the Bacteroides, the Pathomyces, and the Prophylaxis, to name a few. And some of those bacteria have genes that are showing being oligotrophic photosynthetic bacteria, or they are part of the nutrient cycling, or they produce natural products such as anti-tumor and anti-cancer compounds. For example, Bacteroides had genes that are feasible for degradation of polysaccharides. So maybe they were helpful in breaking down the damaged seaweeds or making. And then Prectomycetes mineralized organic matter, and Chloroflexi that were in my study were found to be nitrogen fixation, involved in nitrogen fixation. So looking ahead from what I studied, metabolic coding is a very impressive tool, very strong tool that 5 years ago when I started my study, I learned about microorganisms. However, today's knowledge, the advancements of today's knowledge has led to the discovery of microorganism barcoding to just 1 liter or 3 liter or 5 liter of environmental sample or water samples. So we don't need to use invasive methods or labor intensive methods. So multiple resource to know who is there in the environment. And we can know not just the bacteria, which is small, we can also know the fishes who has passed through a particular water column or a particular marine habitat. So obviously when we learn to learn bacteria, we also want to know what they do. So this is always goes back to the fact of bioprospecting, because we know that the bacteria, if it's helpful, if we can know the pathway by which it's helpful, how they produce a particular bioactive compounds, we usually go to the exploration of these functional genes, for example, the halogenase, which is also part of my study, the bromoperoxidase, because halogenated compounds, in my case, They stabilize or activate the metabolic compound, the metabolic activity of variety of compounds, which can be example used from the CV part. So functional genes like halogenase, dehalogenase, PKS, polypeptide genes, NRPS, the non-ribosomal protein synthase genes. Those genes are helpful in producing those natural products or the bioactive metabolites. That people often wants to study. So using different strategies to target these genes of interest will lead to more discoveries or novel or other important bacteria. So thank you. That's it for my sharing for today. I'm sorry for some of the interruption that happened.
Thank you, Dr. Najat. And are you feeling comfortable, Doctor, or you wanted to, because we understand the situation of having the presentation online from home, there will be a lot of issues as well. So if you want some more time, maybe we can proceed with Prof. Ramanathan's presentation, followed by the question and answer session if the audience wants to ask some questions to you. Is it fine?
Oh, it's okay. It's okay.
Should we proceed with Prof. Ramanathan one, or shall we go ahead with the question and answer session, Doctor?
I'm okay with either one. It's okay.
Okay. Then if that is so, then let's stick on to the flow. I'll open the flow for the question. If any participants would like to ask the question, please unmute yourself and state your name, and please proceed with the questions. I do have a question because it's also stick onto my field, and since you use the NGS sequencing Sanger method, Doctor, from your perspective and from your study, when of course we all know that the NGS is the most strongest method and it will give more diverse bacterial identification. So compared to the culture method, so rather than having a deep study on the culture method, why don't we extend the number of sampling on the NGS itself?
I think it goes back to the objective of research. If your research just wants to find out who the bacteria are, then yes, go ahead, go for the NGS and do everything through NGS because metagenomic data is very powerful. You get to know a lot of things. However, if you're interested in metabolites, especially metabolites, if you want to mass culture your bacteria to produce particular metabolites, you can use the bacteria for biotransformation of those metabolites. Then in that particular sense, yes, you have to go for culture dependent method. If anything else, then yeah, go ahead for culture independent method.
Culture independent method, fine. And in terms of the diversity itself, compared to the culture independent method and the culture dependent method, for sure the culture independent method will have a highest diversity for sure. There's no other doubt about it. But in terms of comparing your studies with the similar sort of studies from other algal group, how do you think that the diversity is higher or lower in the samples that you have targeted on?
Like if you look in a general trend, the general trend is actually similar. However, from my exact small snapshot of study, could compare with other research, and like if you read through, I understand the sampling was like more longer. They took multiple samples from same size. When you compare things like that, then obviously my research has lesser diversity and lesser. But the diversity is still you can say like adequate if you compare like snapshot to snapshot. Other research is like 1-to-1. Therefore, it's fairly diverse because Malaysia water has a lot of bacteria, like of microbes. So if I compare like my microbes in the marine environment of Malaysia, it's similar. The diversity is similar.
I see. That's great. Good information, Doctor. I think it's really good work, by the way. And how much sample did you use for the NGS? You use the water sample for sure, and you use
So I
the
actually use the seaweed sample.
direct
I
sequence.
harvested the seaweed. I extracted the seaweeds.
Okay.
Get the DNA.
Okay. How many samples do you use, Doctor?
I think the total started from 9, and then after we filter, only the 3 was used for the thesis.
Yes. Okay, fine. This is quite common. I understand the situation. And the cost incur for the single NGS sequencing is too high as well, anyhow. back in last 5 years back, it was too high, but now
Yeah.
it's
Now it's actually quite affordable. So yeah, you should
affordable.
like
Yes,
transfer it.
exactly. Thank you very much, Doctor. Thanks for presentation. Is there any other question from the floor? If there is no question, then I would like to thank Dr. Najat for her excellent presentation, and this will be helpful for other participants as well. And with this, I would like to move on to the second presenter, our special invited guest, Professor Ramanathan. I would like to introduce about Professor Ramanathan, and it is really astonishing, and his CV really speaks. He has more than 30 years of experience in teaching and research, has published more than 225 international and national reputed papers, produced more than 34 PhDs, and they are placed in uh, good faculties as well as a researcher, scientist in internationally reputed institutions. And he has projects worth more than $2.5 million US dollars. He has secured a lot of grants and international in collaboration with international institutions, and he has dozens of awards. Some of them are like the uh, energizing Bharat Award from the Bharat Petroleum 2015 for the water conservation in fluoride affected area, and he's also awarded the Huyushi Award from Japan for the Environmental Conservation 2019, and he is also listed as top 2% global scientist by Carbon Analytics. And there are much more things to say, and I think his presentation will speak on its own. I would like to pass the floor to Professor Ramanathan, and thanks for accepting our invitation. Thank you, Prof. Floor is yours.
Thank you, Dr. Akbar, and thank you for the university for giving me this opportunity to interact with you. And I'll be talking. I'm happy to talk today about in the Ocean Day. Can you able to see the slide?
Yes.
Okay. So today I'll be happy to talk in the ocean, the decadal ocean, and the eighth, as you pointed out, is passed out. So on today I will be talking about the mangroves, which are the lifesavers, we can say, and it is mangrove conservation management and carbon benefits. So we have a team. We have been working starting from when I joined Annamalai University. I got inspired from this Center for Advanced Studies, Professor Kadireshan, Kandan, and other people working there, and started to work in Annamalai in the mangroves, and still try to continue to work in this mangrove and developed a mangrove research group in JNU, Jawaharlal Nehru University in New Delhi in this region. So this is, as you know, she has pointed out that that Ocean's 8th is the World Ocean Day, and has been designed by the United Nations as a humanity to celebrate the world oceans, because it is the largest area it covers. Only land area is limited to 29%, and with a pledge to preserve the oceans, because we are abusing the ocean in a lot of sea surface temperature, pollutions, and everything, and the ocean life and livelihoods, and also this nowadays the plastic pollutions. That is what they highlighted, which is a menace in future is going to come. And this is the ocean is the hydrosphere, as you know very well. That ocean covers 31% of the planet and represents 97% of the water of the planet. This is for the information to the students, and this is the marine environment, including the coastal open seas and other things, Arctic, Antarctic, and other, including the fifth ocean, that is the Southern Ocean. And that we have that, archaea and bacteria. See the photic and aphotic zone distributions varies. So the photic zones which we are abusing and they are polluting is going to be affected, which is high productive, and which is having this temperature maintenance and refrigerator. And the world population lives in the 40 to 60% of the world population lives in the coastal area, and then 80% coastal cities. So in the coastal environment, mangrove is a very important ecosystem in this region. So the mangroves are the salt tolerant species. We know that are also called halophytes, I think in Malaysia, I think everybody might have seen, because you are having around more than 5,000 square hectares of 5 lakh hectares of mangroves, and they contain a complex salt filtration. That is the specialty of this complex root system with salt water immersions and wave actions. They are adapted to low oxygen conditions and waterlogged muds, and they can survive in salinity ranges. That is the specialty from 2% to 90%, depend upon that species, which species. The global species, and this they varies from the shrubs to tall trees. And there are three most common found species: the Rhizopora, Mangla, Rhizopora, Racimosa, and Ravisera germanis in the world. But in India and Malaysia, the species will be major species Rhizopora and Abyssinia, but we have the subspecies. But Mangla is also common in the India. Mangroves are the tropical forest colorating, and then with their ability to store vast amount of carbon, that is what we are going to discuss about that. Mangrove forests are the key weapons to fight against the climate change. And special characteristic of mangrove forest adaptation to low level oxygen, because that is the specialty of that, and they take oxygen uptake and the nutrient uptake from their different. There are around 6 or 7 major root systems, and the trunking roots, and then the still roots, and then the nematopores, pencil roots. They are all having a mechanism by which they will take the oxygen and nutrients and survive. And the mangrove forest accumulated with the mud and the dense mangroves is common, but just you are also having in the eastern peninsular Malaysia, and also your Sarawak and other regions. And because Indonesia and Malaysia is having a lot of high diversity mangroves in the world, next only that comes the India, Sundarban, world largest mangroves in this region. So as you know that this mangroves can be happening in that in the fringing area, where this excavated area where the high tides comes in the region, and then the riverine. So in Malaysia and India also we have the riverine, and we have the fringing in the Lakshadweep Islands, and then the basins. During the high tide, the water comes and then they forms at that, and overwash also is there. And dwarf nowadays, because of the climate change and also sea level rises, we get a lot of dwarfed mangrove species, and you will get the species diversity in such a fashion that Abyssinia and Rhizoporas and the mean high water during the spring tides and the mean tidal level. So it depends upon reaches there. So accordingly that it reaches in different places, you will get the different species and different types in that plants. And these root systems, plants remain submerged in the water. The tree species can tolerate high salt concentration. That is what we and the aerial roots for sucking oxygen, the nematopores. That is you get the nematopores in these regions, and this is the still roots. These are the still roots you will be getting, and then the nematopores will be growing in these regions. Mangrove species are specialized about the ground roots called the breathing roots or nematopores, and then they have the pencil size. And also they have many pores through which the atmosphere enters. And then the unique root system holds soil against. That is also a very important component for this root system to survive. And you see that how this sequestering this carbon is happening is there. So we can call it mangrove sediments having the above ground mass and below ground mass and litters and sediments. And the mangrove sediments have high trapping efficiency of carbon. So it is allochthonous from a riverine, autochthonous this litter. So two sources are there. One is the transported source, in situ sources from the trees, phytoplanktons, and benthic epipelagics. And then the mangrove sequesters atmospheric CO2 to form their leaves, stems, and branches. And then the carbon-containing leaves, stems, and branches accumulate the sediments and the roots and bury them, reducing oxygen levels and slowing of the decomposition by bacteria. Buried carbon can remain stored for continuously for a longer time. So the carbon remains stored. That is the very important. That is, it stores for hundreds of years and sometimes millions of years if it is older, and it is released to the respiration a little bit, and by the mostly by the if you deforestations disturbed by the human and the erosion and storm activities like super cyclones and the storm surges happens in these coastal areas, especially in India. If you see, disturbance of the sediment serves the natural events of hurricane, tsunami, etc. And then the carbon releasing of the stored carbon back into the atmosphere CO2. That is the So this is, I know that you know this function of this thing. They are one is a salt tolerant thrives in that we have been discussing, and seed droppings. There is a theory also there's that all the Southeast Asia, from there only the seedlings have been migrated by the sea and distributed throughout the Asia. And the fresh waters from the seasonal runoff reduces the salinity. So we need to have this freshwater flow. There is land-water interaction. That's why Lloyd's program is there. The globally people try to understand the land-ocean interaction. And leaves falls to start the formation of rich detritus. And then the fungal and bacteria, already the earlier speaker has talked about these things, how this seagrass and the bacteria plays a major role in the breakdown of the fallen leaves. And then the crustaceans and other things, they survive. And then the small fish will survive on them, phytoplanktons and crustaceans, and then the larger fish thrives on this system. So if you see these distributions from 20 to 50 species are more dominant in these regions. So especially Indonesia and Malaysia is having that a lot of things, and still there is some issues there also nowadays coming up, like all over the world. The deforestation is a problem, but some of them governments are taking a lot of initiatives, including the philanthropists. For example, the philanthropist in Bombay, you know that already highly polluted water centers and mangroves are destroyed. But the Godrej, that is their family has been taking that Vikroli one mangroves around some 3,000 square kilometer area. They have been maintaining for since 1947 in that area. And also forest departments and MS Swaminathan foundations, they also maintain this. And this is how this mangrove makes the 1% of the tropical forest of the world. And in India, I will be start talking mostly on this Indian side. India has 3% of the total mangrove cover in Southeast Asia, and we have this Avicennia avicensis, Morinda, Rhizopora, Mucronata, and Sonorita alba, Avicennia alba, and then the Heritiera. So we have a little bit of Naipa freshwater species, like in Malaysia you have. In the Sundarbans we have a little bit, because nowadays salinity is changing. That's why Naipa is not coming in this region. And we have this southern states, a lot of mangroves, maximum the world largest mangroves in this region, that is India and Bangladesh. So we have been working in these two regions, and then we are working in Andhra Pradesh and Tamil Nadu and Gujarat. We have been working in this region. So the value mangroves goods have this is that the timber, community level, national level, and the global level. So the timbers and firewood, fodder for animals, traditional medicines, food, local employment, erosional control. That is also very important, especially protection for the storms like tsunami and all. And then the silviculture, shrimp and crab industries, and charcoal productions, ecotourism. So this is what Malaysia, I think, and Indonesia and other countries, even Australia, if you go, they have the ecotourism. In the mangroves. In India, we try to develop in this, especially in this Godavari regions of the Andhra Pradesh. And then the water quality management, you get the eco-hydrology is very important there. The groundwater is getting the fresh water from this, and preservation of biodiversity. And then it is a good indicator of the climate change globally, because if there is a climate change happens, the mangrove is the first indicators. And also I told you the land-ocean interactions. For example, in the Gujarat region, so wherever there is the rainfall is more, that means fresh water is coming. Then you will get the diversity of the mangroves, and the species diversities and density of the mangroves will be increasing compared to that of this. So the intensity of rainfall and freshwater flow. So we need to have the rainfall or the river water to come and compensate that. And you can see that the diversity of the species, that is floras, and also faunas we have, but that is you get that Avicennia marina and Avicennia marina young seedlings in these regions, and you get Sonorita and also Salveda. So these are the species in the different, because they make them very productive in these regions. So as you know that since you are from the institute, you also know that they are the blue carbon is stored. We have the green carbon, blue carbon, gray carbon, and black carbon. So the blue carbon is mainly stored in these coastal areas from these coastal ecosystems. And the carbon sequestration is the process of capturing and storing the atmospheric carbon dioxide. It is one method of reducing the amount of carbon dioxide and reducing the global and climate activities. biotic sequestration is happening in that, and you see how this gaseous loss and decompositions and organic matter, they sequester the carbon, and transfer of atmospheric between biotic and pathogenic carbon fluid. So they store it for years, hundreds of years. So the most important terrestrial carbon sequestration is the forest, soils, and wetlands. So the tropical forests are the good, and all the soils and wetlands are also sequestering the carbon in this region. So tropical forest is having 30 to 40%, at least 20% will be coming in the wetlands of this mangrove ecosystem. You can see that wetlands storing carbon. Every year, the coastal wetlands sequester enough carbon dioxide of the burning of over 1 billion barrels of oil. So we need to have these mangroves to compensate this. If you have the 720 tons of coal emission, it should be offset by 1 hectare of mangroves. So always there is you have to, even if there is a deteriorated, then rejuvenation and replantation you have to do. And the coastal wetlands is the only habit that can continuously sequester and store carbon for millions of years. And 1 hectare of seagrass, she already talked about the earlier speaker, can store about, that's why in marine biology, I think even Akbar has worked, can store a lot of carbon capture. So although it covers the coastal wetlands, less than 1% of the ocean, but they store 50% of the seabed-rich carbon. That is why they are very important. You see this blue carbon storage, the seagrass. It is one is the soil organic carbon, living biomass. So the salt marsh and seagrass, they have the soil organic carbon is more. In estuarine and ocean mangroves, we have both the living biomass and both are storing, but relatively all the soil are carbons. As far as the tropical forest, the difference is that they cannot store much of the organic carbon. The living biomass is having a lot. So similarly, the economic value, if you see that one, they have the fisheries, ecotourism, pollution, energy, wood, timber, carbon sequestration. You can see that one, that is compared to that of the economy, fisheries, ecotourism, and coastal protections. Carbon sequestration is also having the higher values. But only thing, so far nobody has valued this, how this fodder, water bioremediation, protection of salt intrusion, and pharmaceuticals and environmental. People have not valued this thing. The work is going on. Some of the works are available in bits and pieces. And you see that above ground mass and below ground mass, they store the maximum amount. You can see that one, that is the soil below 30 centimeters in this mangrove areas, Indo-Pacific. They store the maximum out of this 1,200 milligram per hectare of the carbon. They store around 600 to 700. So similar compared to the tropical uplands and boreal forest and temperate forest, you see how much it can store in these regions. And you can also see that one in the below ground and the above ground mass in the different mangrove species of India, Malaysia. Malaysia also you see that one, they have the above ground mass and below ground mass studies are available. In India also we have done some work in this region. So the mangrove storage of this carbon is very important, and especially Indo-Pacific region is famous for this because tropical and some temperate regions have the mangroves. Beyond that, we don't have that much mangroves. So the mangroves, this is what I told you, still roots in these regions, in these red mangroves. And you can see that the significant mangrove sequestrations compared to that of the all ecosystems, forest ecosystems, the mangrove ecosystems have the turnover time also is relatively less, but it can sequester more carbon and store the carbon stock in the mangroves. Wherever mangroves are there, you can see that carbon stock is increasing in these regions. So we have been working on this, and then we are working on the Sundarbans from the Indian side and Bangladesh side. We found that the Bangladesh side is having that relatively higher percentage of below and above ground mass organic carbon compared to the Indian side, which has been disturbed except in 1 or 2 pristine core areas. Remaining areas, we have this increases in the region. So similarly, if you see that Pichavaram and Tamil Nadu regions, and recently that in the top few centimeters, you see they lose the loss of carbon in the bottom because of the freshwater flow is reduced in the regions. So we are getting the less amount of this in the past 100 years or so. We in 30, 40 years, we are getting reductions in the organic carbon deposit in this region. So we should also try to understand that health of the mangroves, how they are capable to sequester this carbon in this region. So whether it is coming from the terrestrial plants C3 or C4, and also the freshwater DOC, marine DOC, marine particulate organic carbon, marine algae and bacteria. So by using this ratio of this that and also this model we are using, and then we found out that in the Pichavaram area they have the disturbance of this. They are not getting much of the freshwater DOC in the recent years compared to the Sundarbans in this region. So we should be able to identify that sources so that we can tell that the mangrove is going to still healthy, and what are the interventions we are supposed to take in this rejuvenations or the freshwater watershed management in these regions. So we can see that one concentration of this mangroves in the sediments. If you see that one, so Sundarbans is having a lot of freshwater flow, and they cannot store much on that. Compared to the Pichavaram, they store relatively from lower to higher region, the pristine area, they store maximum organic carbon. And if you see this Godavari region, they are storing the maximum the pristine area. They store a lot of organic carbon. The Orissa also they store a lot of organic carbon in the systems. So significance of the mangrove conservations and carbon benefits. So you can see that one in the Kutch area that only the dense mangroves regions store the maximum carbon in this region compared to the moderate and the spare regions. You see this dense area, even though the Gujarat, the Saurashtra area is the only maximum deteriorated industries are there. And this South Gujarat and Kutch, they're relatively better in the regions. They are having dense mangroves still. And then here, if you see the restored areas in the Bhitarkanika of regions of the Orissa, so there we get this restored area also stored. So this is done by the MS Swaminathan Science Foundations along with this local forest department. Without forest department, we cannot do any work there because they are the one working in the field. And then the restored mangroves store the maximum compared to the at least similar to the dense mangroves in this region. So the economy of this blue carbon is also very important for policy making and mitigation measures, because we should conserve that. That's why we have established several centers. Recently, we have established in India Center for Sustainable Coastal Zone Management from the Ministry of Environment to find out this, reduce the coastal vulnerability and conserve the coastal environment, and increase the blue carbon storage in the regions. And they also, this is the carbon market can attract the government, private stakeholders. Already that is philanthropists and also working in some of the regions, and they will be coming out to restore and the conservation so that the blue carbon credit should be given to them. And then the countries or institutions which wants to emit greenhouse gas can buy credits. So while the country or institution sells their credit, will be awarded for the reducing emissions. So the policies also will be come into that picture. And then the payments for the ecosystem services, what the mangroves do, some of them are not valued. We have to value it, and then we have to collect the money from this for the ecosystem service they pay. And that's how we can conserve this blue carbon storage in the mangrove regions. So how they do the plantations and the restorations of these mangroves, especially in the Pichavaram and this Godavari, that Kuringa mangrove, they first look into the salinity levels of the soil. Mangrove species, Avicennia, Marina, Avicennia officina, this and Exoguera, Agalocha were selected for the planting the degraded areas. Reason being that these species tolerate a wide range of salinity. Generally, in the dry soils in the mangroves, we get around 140 ppt during the summer. To reduce that, the high salinity and tidal flushing was facilitated by constructed canals. Due to this, the salt are slowly leached out and soil salinity is reduced. The reduction in the salinity improved the survival percentage and also reduced the saline stresses of the young seedlings. So in generally in this region, we do it in the October, November, because the return monsoon, that is, we call it as one is the Indian summer monsoon starts in the month of, already it started in the June in Kerala, and all the way it goes to Himalayas, and then it get become blocked by the Himalaya and come back to the Bay of Bengal and reaches Andhra, Tamil Nadu and Andhra. That's why October, November, this we call it a southwest monsoon. during the northwest monsoon, southwest monsoon. During this period, the rainwater reduces the salinity. So that time they put the seedlings in that one, and that the salinity comes to 10 to 15 parts per thousand. So they plant this species in these regions in the different, so that they will grow. 8 month, they generally select the 8 month old mangrove saplings raised in the nursery were used for planting. The mangrove saplings planted along the slopes of 20 to 25 centimeters of the canals with a gap of 2 meters. So the de-sticking of the canals and the casualty replacement also done by them in the two regions. So this is how they do this. The main channel for two channels of this, there are main lot of islets in that, lot of channels are there, main channels. Out of this, they connect the two channels where there is less mangroves in this region, the degraded mangroves or dwarfed mangroves. This one is at 45 degrees and this one is at 30 degrees, they make the angle. So this is like a bonefish setup they will do, so the tidal flushing will take place. And then they identify this so we are also involved in identifying the soil characteristics or the sediment characteristics, where there is more of the montmorillonite clay. So that area will be helpful for the productivity of this and the growth of the mangroves in this region. So this is how they did it in the 1971, 9, and then 2004 and 2004. That luxuriant mangroves goes after this. So this is also happening, I think, in Malaysia. I don't know, salt pan deposits in India because of the salt pan deposits and silvicultures and aquacultures. So mangroves are also degrading in this region. So the clear felling through these are the reasons for this degradation, exposure of the mangroves, evaporation of soil water, stagnation of tidal water, development of traps, subsidence of sediments, especially in Sundarbans, we found the subsidence of sediments because of overexploitation of the groundwater, the land subsidence is taking place, and the development of the hypersaline, and no natural regeneration because it became dwarfed and bald in these regions. So that is how this that evaluation also done. It is having 1,94,000 per hectare annually, the mangrove benefits. And then according to the study, more than 75% of the soil carbon emissions came from the mangrove deforestations. So we have to be very careful, especially where the dense mangroves are there in the Southeast Asia. And we have to be very careful in that one. So the loss of soil carbon from the destruction of mangroves and other coastal ecosystems contribute at least 3 to 19% of the global deforestation, causing CO2 emissions. So mangroves, when it is deforested and cut, and then it is the restoration is not taking place, that will lead to a lot of loss as well as carbon emissions. And already we know that sea surface temperature and air temperature is increasing. And then this the reduction in carbons, they have estimated why mangrove is important. 1 hectare of healthy mangrove 1 year sequesters the CO2 equal to that of the one car driving 16,000 miles. So 1 hectare of the mangrove deforestation will be releasing how much? One car driving 4,50,000. So it releases equal amount when you destroy it. And the counterbalance the carbon loss over 10 years period, we have to carbon 4 hectares of mangrove needed to be planted for every hectare of deforested plants. So if you 1:4 ratio, 4.5 ratio, like in the coastal groundwater, we say that if there is 1 meter of fresh water is over pumped, then it will be having that 4 meter of seawater will intrude. Similar thing that happens here in this region. And then the international agencies, LOA, UNICEF, UNFCCC, and NEPA are also working in this. And then they are developing the policies and the scientific needs. Finally, where their goal is to enhance the conservation of the coastal habitats in the regions. So the steps to conservations of this grassroots efforts to save mangroves from destruction has been taken from the local community and stakeholders. That is what successfully done by the Forest Department and MS Swaminathan and the Cartridge company in India. In some areas, mangrove restoration is underway. UNDP and UNESCO has played a key role. And in India, the National Mangrove Committee of Ministry of Environment, Forest, and because wetlands are relatively now got a lot of attention by the ministry, and a lot of rejuvenation and restoration is taking place in the region. And then the HOPE projects like NGOs are also working in Mumbai along with this Godrej and regions, and the horticulture departments are also working. So there are several agents working in these regions. So similarly in the Gulf of Kutch, state government, and they're planting this, and in the Gujarat and Bombay Goa. So they planned this different, including the municipal corporation, forest department, central government, and then they create a biosphere resource and then prawn and Pokali regions in the regions. So what are the benefits? So characteristic of the mangroves, environmental plasticity of Avicennia marina to tolerate extreme salinity, temperature, and light intensity, and its adaptation to different soil conditions is well proven by different studies. Large trees with complex root systems like Rhizophora species facilitate the deposition of the particles to much larger extent than the trees that are smaller. So when they have the more deposition of the sediments, then that will store more carbon, which is because it is having clay and organic because cation anion exchange capacity by which it will store it. And then there is estimate that organic carbon can 10 to 11% in the sediments beneath Abyssinia species is stored. So each species has a speciality of this regions. So it is important for the view of conservation of the afforestation. So species will be identified according to the salinity and the local soil conditions. And especially Rhizophora abyssinia species are planted throughout the world to store this mangrove and restore these mangroves and then store the blue carbon in the region. So Anthropocene, that is, you know very well that agriculture, aquaculture, but agriculture at least people are not that much affected. But if aquaculture and tourism, if you don't have the proper ecotourism management and urbanization, so this is what the main culprit in the cities like Mumbai and all. And we are having this problem, and then shrimp farming and deforestation because people needs alternate fuels in the coastal areas. So that because the 40 to 60% of the population lives there. So for example, for that only the government of India started the gas distribution freely to them so that they will not cut the forest in this region. So some type of measures has to be taken in this region. And this is how this sea level rise will be impacting. For example, if the sea level rise is there, if there is any sea surface temperature increasing, a convection current increasing, and then the sea level rises, and cities like Lakshadweep, Bangladesh, and some of the regions will be, Maldives will be submerged in these regions. When they started to submerge, you see that slowly they deteriorate the density of the mangroves in these regions. And then ultimately you will get the deteriorated mangroves in these regions. Finally, you will get something, the carbon will be released and eroded and transported by the coastal processes like tides, longshore current, etc. in these regions. Similarly, the tropical deforested shrimp culture is releasing a lot of carbon into the system. So potential evaporations is that coming from the shrimp cultures when they use it, and also the quality of the groundwater and surface water and soil deteriorated, and also carbon will be released. So you have to afforestation program has to take in these regions to counter that in this region. So overall effect of the anthropogenic is one is that, as we talked about, is that anthropogenic phenomenon and human industrial activities, greenhouse gas emissions, and increase of air water temperature. For example, the air temperature 0.5 in 1880 between and 2002, 0.2 degrees increases, then the phytoplanktons growth will be affected. Rising sea level, as I told you that we are expecting in 3.2 meter per year in future, and ocean acidifications. So this is again 0.1 pH unit since the pre-industrial unit is much higher. Relative to the climate warming and sea level rise, ocean acidification as a chemical process often morely strongly affected by the opening impacts. So, and also if there is acidification, a lot of pollution entering the heavy metals that will be dissolved, and then it will be available to the aquatic systems, faunas, floras, and that is affect the entire system in the region. So that is how this anthropogenic impact. So some of the anthropos like our anthropopaths, that is the like COVID situation. So you see that when the COVID situation, the globally, we have reduced the using the electricity in these regions. In India also, if you see that one, that there is a reduction in that month of when there is a happening in the, so the nature is teaching us that if you have this controlled emissions and you have best managed pollution methods and treatment methods, then you can also reduce and conserve these mangroves and ecosystems in the regions. So this we have to learn from this lesson. Nature has taught to us from the COVID that the temporary halting of these activities also rejuvenate the ecosystem in the regions. So even in the water quality, we have observed. I think one book has come out from our side. I think if you have a time, you can go through the book. We have mentioned how this COVID has improved the environmental quality of this air, water, and throughout the globe in the regions. We have observed that in this study, in the Ganges, the water quality improved in a short time. Even now, that is we are working on that. Maybe this is a long-term that is COVID problem. So this time we are expecting better water quality in the Ganges and other regions in the area. So that is also having some other problem. That is the forest department could not unattended forest fires has increased during the summer recently in the mangrove area, pest infection and agriculture because of the COVID. People are going there, and the overgrazing and husbandry is happening. And then there are also a problem like budget cut, transfer of environmental funds to other places. So there is a closely related, the ecotourism is also affected in the countries like Costa Rica and India in these regions. Maybe in Malaysia also that might have been affected. And so we have to have this integration of this, better integration of this policies to enhance the mangrove ecosystem and tropical forest to make the sink for the greenhouse gases and reduce the acceleration of this deforestations and stepping over our natural conservation efforts, reducing the environmental pressure on biodiversity, integrated action on biological biodiversity loss and climate change. We have to do it, and we have to. The European Union has adopted the white paper on climate change, and they have given the global forest cover that we half it. That is what they took the oath by half by 2020 and halted by 2030. I hope that will be happen, and then we can reduce this rise in temperature, and the global forest carbon mechanism has to be improved in this region. So in Malaysia, if you see that one, you are having the better mangrove ecosystems in the peninsular Malaysia and this eastern Malaysia, Sarawak and Sabah. And I think you are having the better management mangrove. I don't know much, but we have visited once when I was there in some conferences. In this Matang mangrove is there. And Malaysia is having the coastline of 4,800 kilometers and with mangroves of 5,77,000 hectares, and Sabah, Sarawak, and Peninsular Malaysia, they have the maximum in this region, have potential to store 530 million tons of CO2. So a lot of potential is there for you in this region. So I think it's since the institute is lying there, you can work on this. So that is Malaysian mangroves are largely of river dominated type of built, but integrated river dominated, tide dominated settings and drowned valley settings and carbonate settings. Like our Lakshadweep, you are also having the carbonate settings. Mangroves are exploited by humans for coastal protections and fisheries like in other countries you are also doing, but you are having better ecotourism. Very often this exploration is unsustainable because of this. This is the study recently done by Wang Shou. And they have also found out the 60% reduction in the Matang mangrove and sustainable management policies the government is doing to restore. That's what they identified in this region. So Malaysia is having a lot of mangrove potential to work on this. And this is the Godavari mangrove. You can see this ecotourism. People can come and walk around like in Malaysia and Matang you have. And then you're also having that some certificate you give or forget that is in the tropical 25 degrees, I think, latitude certificate, Matang area you give. So this is how these channels and these still roots are there in this regions. This is the breathing roots in metapores, Abyssinia. This is the Rhizopora standstill roots in this region. So this is what we do that field work, like you do it all in this. And this is the one I am telling this book recently we have brought out. And I think it covers all the ecosystems. If you have a time, I think, and your university can buy from the Elsevier, you can buy that. Thank you very much. And I think I have taken much of the time, and I hope it will be useful to all of you.
Thank you very much, Prof, for your extensive, informative presentation. It's our pleasure also to read the book, and I have gone through the chapter because I was invited as a
Yeah,
reviewer.
yeah, yeah.
Thank you very much, Prof. It was very good, really. Uh, So we have few questions from the floor, and
Yeah, welcome.
if Faiz wants to ask directly, you can unmute yourself and you can ask. Otherwise, I can read the question. Dr. Faiz? Fine. I will. I'll just read the question, Prof. Maybe you can give your suggestion. It's very hard to attract the people for conservation of a mangrove ecosystem. In your opinion, what are other things that mangrove conservation framework can offer to attract more people, NGOs, or perhaps industry player to get involved in this conservation of mangroves?
One is that, as I pointed out, that the government policy, credit policy, if somebody is involved, like I told you that Godrej is involved, and thus company is involved, you should give that conservation equal to that amount, some carbon credit to them. One is that one, and then you should also educate the people that no, and then livelihood, because if they are cutting the trees, for example, for their firewood and for the roofing in the coastal areas, and you should tell them that you should give them alternative resources like no gas for them, and then cooking gas. So if you are giving the cooking gas connection to the people, they will not cut the forest in that region. And you should also educate them by telling that if this diversity of the mangroves is there, because each of them is having the root system where the siblings of these fishes and crabs and things will come in the high tide, and then they have a lot of litters and retreats in this mangrove because and there is shelter for them siblings. They come and grow. Because they come and grow here, this area become very productive. And also it is having the so we have to educate them that because of this you are getting the fish production. If you cut this, the fish production and your economy will be growing, will be reducing. We cannot tell them that blue carbon is there and carbon dioxide is increasing sea surface temperature. But at least we should tell them that you will be getting the credit and you will be getting the livelihood out of this. All these things, if you tell them, then it will be government and local stakeholders, and then educate them. That is the through NGOs and other things, and then give the remuneration for them. That is the one thing that is we have to do that.
Thank you, Prof. I think we have a similar situation in very small area here in Balok, Pahang Coast, where our institute is located, the same state. Because the village people itself, they don't want the government to establish some sort of resorts and hotels in the region. What happened over the past, we have a papers in a few years back, and when you go, I just went there last a few months back, and they said the water quality has steeply increased, and now the quality of water in that particular region where there is a river and so on, it is a grade 1 quality, just because they have fighted with the government, local government, not to construct any any resorts and so on. So the quality of water has keep on increasing. Similar situation, Prof. And the second question is our Institute of Oceanography and Maritime Studies is located near the mangrove area. In your opinion, what roles can we play towards conservation of mangroves as Oceanography Research Institute?
I think you are the best person to answer, because in your marine biology department, you have rejuvenated the mangroves in that region. So only thing is that you have to identify the soil characteristic, salinity, and the movement of this water, tidal waters in different directions. Wherever there is a dwarfing or the baldness of this, because of the salinity, the soil become bald, and then porosity, permeability reduce. And those areas, and you characterize them, and then try to identify the angle at which you can put the channels in these regions. Involve the students, national I think social service or NSS, we call it as so the students can be involved in the Saturday, Sundays, or in the vacations along with the local community. And then you can grow your siblings, typical siblings in your lab, which can be planted after 8 months into that land. So you will also helping the community and also tell them, demonstrate. Maybe I think when I first went to marine biology, I have been demonstrated if the mangrove goes, how much this the nutrient content will increase and the productivity increase and the fish production will come. Something like the demo or the something you can show to the people how this is going to improve. So I think you can do a lot of things besides your, because I know that you have to publish, you have to get the publications for promotions and all those things. Maybe a certain 10% of the work can be diverted like this. Because when we are there, we try to work with MS Swaminathan Science Foundations. Now we are in Delhi, but we are working with some Sundarban, some local NGOs there in the region.
Thank you, Prof. Yes, I still remember just opposite to the CSMP, there is a
Yeah.
rejuvenation process, and the Prof. Kadiresan group is doing
Yeah,
extensive.
2 hectares of land has been rejuvenated.
Exactly. Okay. I think that I'll move on to the third question, Prof. Thanks for very good research on the mangroves in India. Would like to know how universities at India do conservation approach with other entities such as the governments, citizen science programs, and NGOs as well. I think you have.
Yeah. Yeah, one is that I told you that the government is now encouraging the wetland management with institutes and NGOs. So they are supposed to have one member of this from the university and the committee. When the fund is given to them, they should have this committee so that this research, which is done by these universities, will be incorporated into that. That is what they are doing it nowadays. And already that MS Swaminathan, I told you that they are some type of NGOs, they dedicated some national agencies now supported by the government of India. Similar agencies can be developed as a consortium. Like your institute can develop a consortium of this institute, and then the government can fund it, and you can have a linkage between the society, government, academic institutions, like that. And that make the compulsory if anybody is getting the fund, and they should have some this corporate social responsibility. And also if NGO is getting fund, they should also have some research component from the universities to be incorporated.
Thank you, Prof. I think it answers the question. Followed by this other question, conventional tourism is considered one of the aspects that contribute to mangrove loss. Do ecosystem approach can prevent that?
Yeah, this is something that is everybody is thinking on that, because we need to have the two ways we can think into that. One is that the tourism may be throwing litters and pollutants into the water and then they destroy. Another one we can look into that. They may be getting attracted to this mangrove and then they help in conservation of the mangroves and also give some revenue for the mangroves. So it is up to the managers to attract the crowd which is coming, whether to attract them to conserve the mangrove or to litter the mangroves. So that is how the program should be evolved, involving your social, economic people, psychology people. So if people know the psychology of the people, now how to attract them to go to mangroves rather than just abusing the mangroves.
I see. That's good. Thank you, Prof. I'll move on to the other question by Prof. Jalal from our institute. What are the key threats to the mangrove ecosystem in India in general? What are the best measures taken? Is there any significant approach you have observed in the management of Matang mangrove ecosystem in Malaysia, one of the best managed mangroves ecosystem in the world?
First question is, you repeat, in India, what is
The exact threats, the key threats to the mangrove ecosystem?
key threats. So the key threats in the mangrove for us is the one is that salinity. For you, it is not a problem. You get a lot of rain. For us, only in the monsoon that is coming. So now the salinity is increasing. Like I told you, Naipa. The Naipa species is now completely eradicated because the Bengal tigers is actually the breeding ground near that, because when you go to the field, we used to put the tiger in the front page and the backside face of the tiger, because it will come and attack. When you enter the first time in the field in 2000, nowadays that they are telling tiger is also reducing because Nipah is because it is camouflage with the Nipah. The color of the tiger skin with the Nipah skin, the trunk will be same. So you cannot identify the tiger is there. And then the salinity has disturbed. That's why in Indian mangroves we are reducing the freshwater species. One, and then I told you the pollution. Like in Goa, if you see that one, Goa and Kerala and all, they are reducing the mangroves because of the pollutants entering there. So only during the flushing time they are growing there. Now the government of India is thinking of this NGOs to look into that treatment plants or watershed management in this region, so that the salinity maybe and pollution salinity will be increased, decreased. And the third one is that one does sea level rise. The salinity increases and sea level rise increases, accelerate the dwarfing of the mangroves. There we identify the dwarfed areas, and then rejuvenation by this forest department and other people are taking place. And in Matang area, I still remember lightly because I came in 1996 only. I don't have a chance to come after that. After 2002, I have not come to Malaysia also. So that time we went for a conference. That time I saw it is well managed, and I still don't remember. Yesterday only I just recollect that the Matang mangrove in that area. I got a certificate also. I think one certificate they gave us visiting this to encourage us to know that we are reaching that latitude. I think that is the best managed, and I think you have a lot of scope to improve and store the mangrove carbon storage and attract the ecotourism and generate and involve the stakeholders to understand that they will be going to benefit, not the government of Malaysia is going to benefit.
I think it answers the question. There is one question to Dr. Najatul. Maybe I have to move on to Dr. Najatul. Can the relation, I think the questioner means the relationship of bacteria with the seaweed. Could it assist in seaweed conservation?
Because I am not
Yeah,
working on that,
I,
because you are the right person to answer. But seaweed is having, I told you, the carbon sequestration. They play a major role, and especially marine biology has done a lot of work. Even now, I think your institute's person who left you and started one NGO in Ramanathapuram. I think some. He has done a lot of work on this conservation of this seagrass in the Rameswaram area. As an NGO, they got a lot of fund to conserve it and sequester this carbon and productivity of this fish in the region.
yeah, I think it's Dr. Anantaraman. The same question goes to Dr. Najatul as well.
Okay, thank you for the question. Yeah, of course, the bacteria can, that's when we work in a framework, in the research framework, yeah, the idea, the philosophy behind every research is there's always an objective to achieve. So in my framework, yeah, I believe that I wanted to identify the bacteria that can help in the seaweed conservation, seaweed farming, and such and such. And this ideology can be applied to any other marine habitat. So moving on from the seaweed, I am also applying into looking into the NGS or the bacteria identification in other marine habitats, such as for now the corals and others are also including the seagrass. So by knowing which bacteria is there, based on the environmental samples that you take, then you can learn more on those bacteria and how to use the bacteria or how to work on the bacteria in order for the conservation. So thank you for the question.
Thank you, doctor. I think it answers the question. I have one doubt to ask to Dr. Prof. Ramanathan. Back in India, for example, Prof, if we use here for the corporate social responsibility funding from any company, if we spend on it and if we have a very good receipts and so on, we can apply for the income tax reduction. So is there any similar method that India is following? I really do not know because I've never paid the income tax to India, by the way, so I do not know. So if you could, if the government wants to start some sort of activities whereby if the CSR projects money that is given to the local community, if the company can apply for the income tax reduction, it would be a great one. Is there any initiative on this?
No. It is compulsory nowadays. See, we have this environmental impact assessment committee in Ministry of Environment, and then they have the percentage, 5% of that CSAR goes, And then there is an environmental conservation plan also is there. 2% they have to give.
Meaning 7%. Okay,
Yeah.
fine. They are willing to give so, but how about their income tax reduction for the
Yeah,
company?
yeah, they will be completely free from income tax.
Perfect.
Not only reduction. If they give it, they have to show it that CSAR local community is benefited and then environmental conservation plan. If they disturb a wetland, they have to rejuvenate the wetland. If they disturb the water body, they have to recycle the water body. And then using this method. So if they don't do it, that comes under violation. If that comes under violation, they will be fined 10 times of the CSR amount.
Oh, okay. That's great.
This is what, but on paper it is there, but 50% is working.
50% is
People always elude by bribing some officials and all, but 50%, because we are also in the violation committee, we found out that each state is bringing to our notice, this company is violated. They have not spent this money.
I see.
That is also coming up to us.
Perfect. That's really great. I think it is also the similar situation in Malaysia. You do not know about the violation stuffs, but for the income tax reduction, it is considered. I know it. Is there any other question from the floor? If there is no question, I think we would like to thank both the invited speakers, and your presentation is highly informative, Prof. Ramanathan as well as Dr. Najar, and we will further invite you for another presentation very soon. Thank you very much for your time, and also all the participants, thank you for your time and thank you for joining us in this webinar, and we will invite you again to the next webinar next month. Because we will have webinars every single month specifically on some aspects. So thank you very much. With this, I would like to end this session with Tasbih al-Kafara. Thank you very much, everyone.
Okay, thank you
Thank
very
you.
much.
Thank you very much. Thank you very much.